Malaria, a deadly disease claiming the lives of over 600,000 people annually, has been a global health challenge for far too long. But what if I told you there's a remarkable breakthrough on the horizon that could transform how we prevent and combat this devastating disease? Dr. Omar Akbari and his team have achieved the seemingly impossible. They've developed innovative techniques for editing malaria genes within mosquitoes, opening a new chapter in the fight against this ancient scourge. Coming up on this episode, malaria is still one of the deadliest diseases in the world. An estimated 619,000 people died of the disease in 2021 alone. It's a massive amount of people that died due to it.
Which is unacceptable, in my opinion, in our modern age. These chemicals that we're just releasing in mass in the environment are harming the environment. They're killing other insects that are beneficial to us. And so we really want to reduce our reliance on these harmful chemicals and develop technologies which are scalable that can be used.
Welcome to a groundbreaking episode of Future Here. I'm your host, Joel, and today we have the privilege of diving into a world-changing discovery. Our guest, Dr. Omar S. Akbari, is a distinguished professor at the University of California, San Diego, whose research is paving the way for a brighter, healthier future. This discovery has the potential to revolutionize the way we approach not just malaria, but numerous other diseases carried by insects. In today's episode, we'll explore the science behind this groundbreaking research, its implications for global health, and the journey that led Dr. Akbari to this momentous breakthrough. Get ready to be inspired and informed as we delve into the world of CRISPR gene editing and its profound impact on disease prevention. Hi Omar, welcome to Future Here. Glad to have you on the show. To kick off, could you please introduce yourself?
Hi Joel, great to be here. My name is Omar Akbari. I'm a professor at University of California, San Diego, specializing in cell and developmental biology.
Great. And can you tell a bit about your background and how you became interested in science and tech?
That's a great question. I've always been really interested in science from a super young age. Since I was a little kid, I was doing experiments in my backyard all the time with just random creatures I could find, dissecting frogs and things like that. And, in school, I always kind of was really interested in both math and science. I probably was better in math actually than I was in science, but I ended up sticking with science.
And how did this start to shape your perception of the world, how you see science and maybe your part in researching science professionally?
Yeah, I think, in school, I really focused on both math and science. And I was admitted to a magnet high school in Las Vegas that was called the Academy for Math and Science. And there I really kind of focused in on those two topics mostly. And when I went to college, I got a bachelor's, master's in biotechnology and from University of Nevada, Reno. And during that program, I was required to work in a laboratory basically throughout the entire program. And so I worked in probably over 10 different laboratories on campus and off campus. And I really got a really broad exposure of all different types of research going on from cancer to genetics to molecular biology, even vector biology. I worked as a public service intern at the Washoe County Health Department Vector Control Unit. And I did that for 4 summers. And it was a really great experience and exposure to vector biology. And I got to do all aspects of it from field work, going out and looking for pest problems and treating those problems with insecticides, to doing aerial applications with helicopters, to rearing my own mosquito fish and having my own sentinel chicken flocks where I could go around and feed them once a week and take blood from them and test them for viruses. And so I got a really, really broad exposure during my college years.
Sounds really intriguing. And how did you then become interested in CRISPR gene editing techniques and science, and how did that lead to your current research work with curing malaria?
Yeah, I think So during college, when I was working as a public service intern, I saw like a real need for developing a better approaches for managing vectors. And I did this because I was actually, going out firsthand and looking for the problem and treating the problem with the current tools that we had, and realizing how expensive and ineffective it was, and how it, one had to go back every season or basically every week to do treatments, and it was really expensive and costly. And I knew that, while this was effective, maybe in Reno, Nevada, but this probably wouldn't be an effective approach in other parts of the world that don't have those type of resources. So I knew there was a need. And when I went to get my PhD in cell and molecular biology, I specialized in genetics. And that's where I made my first transgenic animal. And that was one of the coolest things ever to do scientifically for me, was to actually inject embryos and make a transgenic animal. And in this case, it was Drosophila melanogaster, a fruit fly. But just knowing that you made this animal, you clone the DNA, and you get to sort of rear it and see the genetics of the offspring and how your technology works, was super fascinating to me. And so I knew that I had the ability to do that. And so when I finished my PhD, a postdoc opportunity opened up at Caltech with Bruce Hay, and this was to develop a gene drive system that they had just invented in flies, and they wanted to transition it to mosquitoes. And I felt like this was a perfect opportunity for me to take my skill set in genetics and molecular biology to Caltech and focus on mosquito genetic biocontrol. And that's where I, moving to Caltech, this was a really big experience for me, coming in and basically setting up a mosquito insectary, kind of being the mosquito person on campus at Caltech, and being able to collaborate and learn from many really innovative scientists, and basically pushed me to be kind of really creative and to be forward thinking, and to try to develop technologies in an applied sense that could actually make an impact in the world. And so that's kind of what led me to where we are now. And with CRISPR, CRISPR was really invented back in 2011, 2012, when the first papers came out on it. And that's when I started working on it, when I was still at Caltech, actually. And we were developing gene drive systems and all kinds of tools that could be useful, and just really testing the power of CRISPR. And so once we kind of got our hands on it and started working with it, we realized how effective it really is. We could actually, for example, in a mosquito, we could encode Cas9, which is the endonuclease that cuts DNA. And then in a separate strain, we can encode guide RNAs. And these are the things that kind of tell Cas9 where to cut. And you could design those guide RNAs to target genes that give phenotypes, like, for example, a yellow body or a white eye. And if you simply cross those two strains together, the Cas9 strain to the guide RNA strain, then the result is all the progeny will be yellow body or white eye, all the ones that inherit the transgenes. And so this was really exciting for us. And this is something that we have replicated in many species and have been using as our tool for developing population control technologies.
Can you discuss CRISPR in general for people in the audience who aren't familiar? The now Nobel laureate Jennifer Doudna has been on multiple podcasts discussing the technology, and it must be quite influential in your field in terms of what it can do, and with relative ease, I suspect.
Yeah, CRISPR stands for Clustered Regulatory Interspaced Short Palindromic Repeats. It's a component of the bacterial immune system, and it can cut DNA, and it can also be repurposed for gene editing or even RNA editing. And it basically just acts as a pair of molecular scissors. And the Cas9 is really the component that makes the cut, and the guide RNA basically directs the Cas9 where to cut. And you can program it to basically target almost any gene. and you can multiplex it. You can target multiple genes, and it's fairly specific and it doesn't have too much toxicity either. So it's been really powerful for all the tools that we've been developing in our lab.
So do you see that this is going to start, or has it already started, a new phase or era in biotech?
Oh yeah, I mean, I think CRISPR is definitely being used for a ton of different applications. Of course, human therapeutics is kind of something that everyone is really excited about. In addition to diagnostics, it can be really powerful for, diagnosing at, RNA at really low levels, and many diagnostic companies have been launched in response to that. But, for genetic biocontrol, which is the field that I'm working in, it has shown a lot of value for developing a ton of different technologies. And while none of these technologies have been really deployed or tested in the environment to my knowledge yet, I think in the coming years, we're going to start seeing these things tested. And I really believe many of them will actually work. And so I think the coming years are going to be really exciting for genetic biocontrol and using CRISPR in a really scaled sense, where you're actually going to scale up your intervention and release it back into the environment to make an impact. And so it might be, I think, for genetic biocontrol, the use of CRISPR might be the first really scaled approach where you're going to see CRISPR being released at large scale back into the environment to make an impact.
Sounds great and very promising. Well, what about malaria specifically and your work regarding CRISPR gene editing malaria? That's the article I found you through last summer.
Yeah.
But while doing background research for our discussion, I read that malaria is still one of the deadliest diseases in the world. An estimated 619,000 people died of the disease in 2021 alone. It's a massive amount of people that died due to it. Can you discuss why you're now utilizing CRISPR specifically for malaria, and how does this work in practice?
Yeah, like you said, in 2021, 619,000 people died, right? Which is unacceptable, in my opinion, in our modern age. What that tells you is that the current technologies that are being utilized are not effective enough, right? They do work, but they're not working well enough. And so we need to develop new technologies that are scalable, that can, basically eliminate the burden that this is putting on humanity. And so this is one of our passions. And I think CRISPR has the potential to be one of the key tools that can help us get to elimination. And so the main tool that's being used to combat malaria is going to be insecticides, right? And one of the underlying problems is that these insecticides that are being used are becoming less and less effective. The mosquitoes are evolving resistance to the insecticides, and so what's happening is we have to keep developing different insecticides and mixing them together, and mosquitoes are still evolving, and what also is happening is that these chemicals that we're just releasing in mass in the environment are harming the environment. They're killing other insects that are beneficial to us, and so we really want to reduce our reliance on these harmful chemicals and develop technologies which are scalable that can be used. And so what we focus on is trying to essentially re-engineer the mosquito in such a way that it will kill itself off. And we have developed a number of different technologies that can do this using CRISPR. And so we really want to try to take these technologies forward. And our thinking is that we would develop, factories that could mass produce our genetically modified male mosquitoes and release male mosquitoes, because male mosquitoes don't bite people, they don't transmit diseases. Only the females actually bite, because the females, basically take in the blood and they use that blood to produce their eggs. So what we would be doing is instead we'd be releasing male mosquitoes as eggs or as larvae into the environment, so they can just hatch out and they can pass along our genetic machinery and basically suppress the population over time and eliminate the burden without having to go out and manually spray insecticides or chemicals into the environment, which are probably harming us in addition to the environment.
Sounds really interesting. And as you mentioned, I read that you publicly commented on your research in an article or press release that your team's research is that this technology has the potential to be the safe, controllable, and scalable solution the world urgently needs to eliminate malaria once and for all. Now, how close to this do you think that you actually are in practice?
I think we are really close to this. In the laboratory, we have demonstrated efficacy for our technologies. One of the difficult things for me and for the lab is that we're just a small lab. We have about 10 to 15 employees in the lab and then some students. So we're not like a biotech company. We're a small lab. We're developing innovative technologies using genetics and CRISPR, and we can demonstrate efficacy in our lab and our insectaries and show these things can work. The difficult thing is actually, how do you take something that you've developed at small scale and actually scale it and develop the infrastructure and capacity in the location that desperately needs it? How do you transition your technology? And the real thing is, the real limiting thing is going to be the funding. Somebody has to take on the funding to build the factory, to build the capacity, to obtain the regulatory use authorizations, to do the field trials. There's just a huge number of steps that need to be taken that become somewhat rate limiting, and so we're really looking for partners to help us, take our inventions and actually scale them in such a way that we can actually, eliminate malaria locally from certain regions and then eventually scale it broader than that. And so that's what we really are looking for right now. And that's been a little bit of a challenge.
What about the genes or regions of the mosquito RNA that you are targeting specifically with CRISPR, and how did you find this? Can you kind of talk us through how you made the findings that these are the genes that have to be edited to get rid of the actual malaria?
That's a great question. So a lot of the targets we target are genes which are essential for the mosquito, and these can be genes that are essential for female viability, female flight, olfaction, like females' ability to find their hosts. It can be genes important for fertility. Genes that are involved in sex determination that can convert females into males. And so these are the kind of genes that we look for. And the way that we find them is by mining the genome. So we've actually been part of the genome sequence of the mosquitoes. We've also took part in large-scale transcriptional profiling efforts. So we've done a lot of bioinformatics to identify our targets. And so we basically have generated the baseline data that we need to actually go forward with our targets. And we've done a lot of really cool things. In the mosquito, for example, we've been able to make mosquitoes by targeting genes using CRISPR that are important for eye development. We were able to make mosquitoes have multiple eyes, like 3 eyes. We've been able to turn mosquitoes' body color from black to yellow. We've been able to make mosquito females, not able to fly, but the males fly perfectly fine. We can kill the females. We can sterilize the males. So we've actually demonstrated all these different things we can do using CRISPR, which is quite powerful. And so we don't have difficulty in finding targets. It's really just validating our targets are doing what we think they do, based off of conservation across different species. For example, if it's a gene involved in sex determination in a fruit fly, is it also involved in sex determination in the mosquito? The only way to know that is to go and target it and to see what that effect has on the mosquito. And so we do a lot of that kind of basic work that then informs our applied work. And one other example I'll give you is we recently had a paper where we were trying to engineer a system in the mosquito using CRISPR. And this was actually a CRISPR RNA targeting system called CasRX. And we were trying to actually encode this system in the mosquitoes such that when the mosquito gets infected with a virus, a specific virus, the mosquito would die. And so we essentially kill the mosquito upon infection with a virus. And we were successful in this. And we showed that if we encode a CRISPR-Cas13 system in the mosquito, when it's not infected with the virus, it's fine. But then when the virus, when we infect the mosquito with the virus, then these mosquitoes have really high fitness effects, and many of them die off from this because of the collateral activity that's akin to CRISPR RNA targeting systems. And so this is one of the first examples of actually killing a mosquito when it's infected with a specific virus, and that was all done using CRISPR. And so this is just another example of how powerful CRISPR can be.
Definitely seems very powerful if you have done all those iterations that you mentioned. Going into the main challenges of introducing CRISPR-modified mosquitoes into the wild populations, what is your team's main suggestion for which iteration you would try to introduce into wild populations?
A lot of my background work has been done developing gene-drive systems. And so we have developed a number of gene-drive systems over the years, probably over maybe 10 different approaches. And so we've actually been kind of one of the front-running labs in terms of that approach. And what I can say is that there are other labs also developing CRISPR gene drives, and CRISPR gene drive is really the system that everyone is really excited about and wants to take forward because of the fact that it can scale itself in the environment. You release a small number and they self-scale, and you can use it for population modification, where you release a small fraction of individuals and they take over the population. And the goal is that, once it fixes, or once every individual has a copy of the gene drive, then they won't be able to transmit malaria, for example, or for population suppression, where once it fixes, the population declines because there's no more females that are available. So those are the two approaches that are really exciting that people are talking about, and they're really heavily funded at the moment. And one of the limitations is that both of those approaches rely on releasing a technology, a CRISPR gene drive, into the environment that you cannot control after you release it. So you release a small fraction of them, they spread, and once they start spreading, you can't stop them from spreading, and so they're going to spread beyond borders, beyond boundaries, and that could be really problematic for understanding the risks, for getting regulatory authorizations. Is it ethical to release something that you don't really understand the risks for, and there's no way to really measure the risks in the laboratory? And once you release it or once you deploy it, you can't recall it. That's kind of what the discussion is. And I don't really find it ethical to do such a release. And so what our team has been streamlining and developing are CRISPR-based approaches that can be used for genetic biocontrol, which are safe, can still be effective, which are still scalable, which can be used now. They will be able to achieve regulatory authorization because they are self-limiting. You release them, and they're not designed to persist and spread in the environment. And that's how we designed them. And we designed them that way for safety. We don't want to release something that we can't control. And so many of our technologies are built with that in mind. And my personal opinion is that I think with CRISPR and deploying CRISPR into the environment, we need to start off by first building the capacity, the infrastructure in regions that don't have the infrastructure. And for example, in Africa, there really isn't a place where you can mass rear and scale such a technology yet. So we need to develop that somewhere in Africa. We need to teach and develop the capacity there. And then we should try to scale an intervention and test it in the environment in such a way that's safe, right? And you want to start off with something safe. So start off with a CRISPR-based approach that is safe, that's not going to spread, that's going to also be effective. It may cost a bit more, but at the expense of safety, right? And so this is kind of our thinking on this, and we have developed at least 3 different technologies that are ready to go to do this. And so we're really hoping that we can, partner with a foundation or with a venture capitalist or investor or somebody that can help us actually take our technologies forward, reduce our reliance on insecticides, and really start saving lives now.
Any kind of unexpected outcomes or side effects that you've already noticed during the lab experiments?
I think our biggest unexpected outcome really was just the phenomenon which we called lethal mosaicism. And so it was something that we discovered. And what we found was that when Cas9 is genetically encoded in an animal, and it's expressed both in the germline cells and also in the somatic cells, so the cells that make up the body at high level, it's not toxic to the animal. So you can actually, homozygous, and you can raise that animal just fine. And then if you make your guidernate line that targets a gene that's essential, that line's also fine and healthy. The guidernate does nothing without the Cas9, so you can raise them separately. But then when you cross these two lines together, you can get basically 100% penetrance. So what that means is every single individual that survives that cross will show the phenotype that you're looking for. And the reason for this is the fact that the CRISPR becomes active in the cells, and in each cell you get a different repair outcome happening. And so the animal is a mosaic animal, but when you're targeting an essential gene, there is a threshold that if you target enough of the alleles and you get above that threshold, which isn't hard to do, then you get the perfect penetrance of your phenotype. And so that was something that was really unexpected to us, because that's not normally what you would see in genetics. And so when we saw that targeting many different genes and many different species, and it was very reproducible, that's when we basically decided, okay, well, this is something that we know we can do, we know how to do, it's very reproducible, and so let's use this as the baseline mechanism for all of our systems going forward. And so our lab is, we are an engineering lab, and so what we do is actually our process is to design something, then build it, and test it, and learn from it, and then go back to that drawing board again. So once you learn, you go back and build something else from what you just learned. And so you keep doing that over time, and eventually you end up with something that is really amazing. But what we've learned is that you always have to kind of start off with your own data. And not that other data is wrong. It's just that sometimes there's nuances that may not get reported or that are not really understood. And so you have to kind of build something and rigorously test it and learn from it on your own. And then you go back and do it again. And with this, we've been able to build so many different systems, and it's really exciting actually. And postdocs in the lab, they get really passionate because they start to see, something that they built, they know how it works, and they know what the next step is already. And so it's really fun to do this type of engineering when you're dealing with building species.
Continuing on the ethical considerations when modifying the genes of an entire species, what is the lifespan of a typical mosquito that carries malaria? I mean, you must be able to go through generations of mosquitoes fairly quickly, right? And how fast do you then expect this to be adopted by a whole population of mosquitoes?
So the generation time really depends on temperature. But in our laboratory, we keep temperature high and humidity high. And so we can get through a life cycle in about 2 weeks in the lab in a controlled setting. And so when you have a 2-week life cycle, you can really scale something quickly in the environment. And some of our technologies, we use mathematical modeling to predict how they'll behave in a population. And what we see, within 10 to 15 generations, we can achieve like sustainable suppression that can last years. And so when you have a 2-week life cycle and you multiply that by 10, you're talking about 20 weeks to 20 to 30 weeks. You're at 10 generations in the environment, depending on temperature. And so, within that short time frame, you can actually achieve like suppression, and that's sustainable for many years. And so it actually goes really quick.
Do you think that it might have any radical effects on the whole food chain?
One of the misconceptions for mosquitoes is that there's actually there's about 3,500 species of mosquitoes on Earth. And really only a small handful of them transmit diseases to us, like around 20 or so. And what we want to do is we want to target the most deadliest ones. So in Africa, the most deadly mosquito that transmits malaria is Anopheles gambiae. And the new one that just invaded is Anopheles stevensii. So those are two that we really want to target immediately. And for dengue, chikungunya, yellow fever, and Zika virus, all of those are only transmitted by two species of mosquitoes, Aedes aegypti and Aedes albopictus. So those are two that we really want to target as well. And then avian malaria is transmitted by Culex Q. That's another one we want to target. So we have about 5 species that we're working with in the laboratory right now that we want to target. And so if you think about how the impact will be on the ecosystem, if you just got rid of those 5 species, I think it depends on, location. For example, like in California, we have Aedes aegypti, which was introduced in 2011, 2010-ish, and it's actually spread throughout California. But if you think about it, it's only been in California for 10 years, roughly a little over 10 years. And so if you remove just that one species from California, do you think it will have an ecological consequence? Probably not, right? And, same thing goes for, Anopheles gambiae and Anopheles stevensii in Africa. These species, at least Anopheles gambiae, has been there for quite a long time, but there are other species of mosquitoes there too. And so if you just got rid of that one vector, their prediction is that wouldn't have such a huge impact on the environment because there are still other mosquitoes that are there that, aquatic animals or aquatic insects or birds, bats can feed on. And so you really wouldn't be disrupting ecosystem too dramatically, in my opinion. But this is something that we need to actually do and test. We need to go out and demonstrate eradication and see what the impact may be on an environment where that species is native, and to see if it actually has any kind of noticeable ecological consequences. And my guess is it probably would not.
Thanks for the answer, and that really puts it in context. What about any regulatory or governmental hurdles your team has faced or anticipates facing?
We have discussed our science with regulators, and we don't see regulating a technology that is safe and confinable, self-limiting, to be an insurmountable task. I do see it potentially being insurmountable if someone were to try to go get a regulatory authorization to release a gene drive system. I think that would be a very difficult challenge to achieve. However, I think we've already kind of thought through this, and we have developed things that can be used now. And so we're actually not at the point of obtaining, regulatory use authorizations at the moment for any of our mosquito stuff, but we have actually developed a CRISPR-based population suppression system for agricultural crops, and we developed a system in Drosophila suzuki, which actually has been licensed by a company called Agrigene, which is based out of St. Louis, and they have been doing field trials. And so they're at the point where they're doing field trials, and with the data they generate, I imagine they're going to take that to regulators to get an experimental use authorization. So I think the technologies we are developing are moving forward. It just takes time, and really with the mosquito stuff, we really just need a partner that can help us transition it and start all these discussions on getting regulatory authorizations and doing field trials and all this kind of stuff. So we're kind of at a point where we're ready to do that, but haven't got there yet.
In a broader sense, how do you see the future of genetic editing in disease eradication beyond just your work with malaria editing?
I think with gene editing technologies, there's going to be a ton of medical breakthroughs. We're going to see like gene therapies. We're going to see a lot of use in agriculture. I would imagine a lot of crops that are being gene edited in some way to produce higher yields or have resistance to pests and diseases. This could help increase our global food security. I think all that is in the works at the moment. And I know there's a number of different clinical trials that are underway for different types of CRISPR base editors or prime editors, and it seems like they're showing promise. So I think over the years we'll start to see, more of that, and perhaps also combined with maybe better ways to deliver the machinery to the cells that you want them. So like for cancer, how do you get the CRISPR machinery just to those cancer cells? Those type of questions, I think, are things people are working on. And so we'll start to see better types of delivery mechanisms and maybe even better editors, right? We have a few CRISPR systems that have been developed, and some of which have been optimized for base editing or prime editing. But maybe we'll start seeing more work in terms of increasing efficacy and specificity, reducing off targeting and things like that. So I think there's going to be just a lot of new development that we're going to see over the next many years, and hopefully some real drugs that we could potentially take that can help treat certain diseases and disorders.
How might the rise of other technologies like neural networks or nanotech intersect or accelerate CRISPR research?
Yeah, I think just maybe providing better targets for genetic diseases like Alzheimer's or Parkinson's disease, maybe using like neural networks that could help with nanotechnology. You might be able to develop new devices that can help deliver the machinery, right? Like maybe they'll develop a small device that you can implant that could, maybe help dose it or get it to the right tissue and cells that actually need the editing to happen at the right time. These type of things that could be developed in the future.
Where do you see CRISPR tech and its societal impacts in 20 years?
It's really hard to predict. I mean, I think we're going to start. I think you're going to walk into a grocery store and you're gonna see a bunch of new crops there that are edited in some way that you're going to be buying and consuming. That's probably true. I hope you'll start to see less mosquitoes out in your backyard because CRISPR, mosquitoes have been deployed and eliminated the problem, and so you'll start seeing less disease burden and people just living happier lives because they're not getting eaten alive by mosquitoes when they walk outside their door, because global warming is actually increasing the range by which mosquitoes survive, and also helps to accelerate the life cycle to produce more mosquitoes and transmit more disease. So I think you'll start seeing less mosquitoes. You'll probably see better crops. You might have therapies for genetic disorders that we don't have therapies for today. So people will be living happier lives. That's kind of my prediction. Yeah.
Those are major impacts right there already. So how do you envision CRISPR's legacy as part of scientific history?
I think CRISPR's legacy is huge. I mean, we had the ability to read DNA, and at first, we got the structure of DNA, and then we're able to read DNA. We're able to synthesize DNA. Now with CRISPR, we're able to edit DNA and RNA. So that ability is huge because with sequencing we can determine mutations that give rise to certain genetic diseases, but we really never had the way to go in and correct those base pair mutations, and now we do. And so I think it's a huge impact just to that, but then also to all the other things that we discussed that it could be useful for in terms of genetic biocontrol or developing better crops, or all these different approaches that are basically underway. So I think it's revolutionized science, and almost everyone that I work with or know is using or working with CRISPR in some way, and so I think that's just going to continue to accelerate.
Sounds really interesting. What about as a piece of personal advice? What advice would you give to young scientists or students looking to enter the world of CRISPR research?
Just that, CRISPR is just a very powerful tool that can be used for many different purposes, and just to, use it carefully and wisely and remember the ethics all the time. There are certain things that are considered off limits in terms of CRISPR, like, for example, editing the human genome, for example, or the germline. That's not something that is considered ethically right to do, and it may never be, right? And so just keeping that in mind, understanding, what the possibilities are, what the limitations are, what people believe is acceptable and not, and always keeping ethics, at the forefront when you're thinking of doing things, I think is something that is really important.
That's really good advice. Who would you like to know more about your work?
We would like people that are, involved in sort of funding, like, for example, philanthropies. We would like them to know and understand our work and how our work differentiates from, other work like gene drive, which may not be something that one could use anytime soon. In addition to investors, I would really like Elon Musk to know what we're doing. For example, I think I know he's really focused on space travel and other things, but I'm sure he's been bitten by a mosquito before, and he understands that they transmit diseases, and so if there were additional resources that could be used to save lives here on Earth, I think Elon definitely has the resources to do that, and I think having him know and understand what we're doing with engineering mosquitoes, I think, could open up new opportunities for us.
Lastly, do you think that humankind has the ingenuity to solve some of our biggest environmental and scientific challenges?
I do think there is ingenuity there. And I would say in regard to our work, part of the reason we chose to work on disease vectors and pests is that we really do think that, if we were to use new innovative technologies, that many of these problems are solvable. Like, for example, the mosquito problem, you really just have to prevent the mosquito from biting people, right? And yes, it's hard because, in some places there's a lot of mosquitoes. And I was just in Yosemite a few weeks ago on a hike, and I walked through this area and I was fully clothed. I mean, I had pants on, hiking boots, a long sleeve shirt, hat, and there was just an area we walked through that was just infested with mosquitoes. And, I looked at my shirt and there was about 10 of them on my sleeve, and they were biting through my shirt, and I just ran. I didn't have anything else I could do at that point. And so, that's an example of, even though you have the knowledge, but the problem is, I didn't have the right tools. I didn't have the right clothing on to prevent them from, biting through the shirt. And so I think with the new tools like CRISPR that are being developed, if we use them right, we do have the ability to actually solve this problem. And I would argue that this is one of the biggest problems on Earth. We have more people. Actually, mosquitoes kill more people on Earth than any other animal, and so that's a huge problem. I mean, you have less people dying from war than you do from mosquitoes. So we really need to go to war on mosquitoes, and we need to take on new technologies, and we need help to scale our inventions to actually, reduce our reliance on all the other approaches we're using that aren't working anymore.
And it's a staggering fact. I had a look at one of your previous interviews on YouTube, that it is surprising that the extent of fatalities is that large due to mosquitoes in the world. So it's not an intuitive thought for many people, I guess. Thanks so much, Omar, for the interview. I really enjoyed our discussion, and I hope you all the best with finding the partners for your lab team, and keep us posted and the general public posted with your breakthroughs in CRISPR gene editing.
Great. Thank you so much. I likewise. I also enjoyed the interview, and I wish you the best of luck on all your future interviews. And I'm looking forward to seeing this one on YouTube when it comes out.
What an incredible conversation about the potential of CRISPR gene editing with Dr. Omar S. Akbari. The potential of gene editing to combat malaria and other deadly diseases is nothing short of awe-inspiring. It's moments like these that remind us of the boundless power of science and human ingenuity. If you found today's episode as captivating as I did, please take a moment to like, share, and subscribe to our podcast and YouTube channel. Your support really enables us to bring you more enlightening discussions with visionaries who are shaping the future. Malaria may have claimed countless lives, but Dr. Akbari's work offers a ray of hope that we can finally turn the tide. Imagine a world where we can prevent diseases at their source, saving lives and alleviating suffering on a global scale. It's a vision worth supporting. Together we can look forward to a future where deadly diseases like malaria are no longer the threat they once were. Until next time, stay curious, stay inspired, and keep exploring the limitless possibilities of science. This is Joel signing off from the Future Here podcast. See you in the next episode. Bye.
Future